Okay, we'll get started. Hello, thank you for joining us for today's webinar on fundamentals of iRECIST and imaging endpoints. My name is Amanda Weber, Business Development Representative at Merit. For those of you who are attending for the first time, we're delighted that you could join us. And of course, we extend our deepest appreciation for all returning attendees as well. In this webinar, we will be discussing the principles of iRECIST, the information needed to collect imaging endpoints, as well as special considerations while using the iRECIST criteria. If you are joining us live for the first time, you may also view our previously hosted webinars on our website at www.meritcro.com. Under the Resources tab, select Webinars to replay your on-demand webinar of choice. We also host this content on our company's YouTube channel. Please note we are recording this webinar so that it can be added to both our website and YouTube channel for replay. All attendees will receive a follow-up email with a link to this recording, which we do welcome you to share with fellow colleagues and industry connections as you see fit. All participants will be muted during the presentation. However, we will devote time to questions and answers at the end. We do invite and encourage you to submit questions at any time throughout today's presentation via the Zoom Q&A functionality. Dr. Kim will address as many questions live as time permits, and then all remaining questions will be answered directly via email following the webinar. Without further ado, I would like to introduce our speaker for today, Dr. Gene Kim. Gene is Merit's clinical scientist. He implemented the iRECIST and other reading criteria into our Excelsior software platform, delivering automation in a single system for all image and workflow management. Please welcome Dr. Kim.
Thank you very much for your introduction, Amanda, and thank you for everyone who's joining our discussion today. Today, we'll be going over the purpose and the background of the iRECIST briefly, and we'll be touch basing on a measurability of the lesion per iRECIST recommendations, also the lesion category that is used in iRECIST, lesion response criteria, and then we'll be also talking about the trial outcomes, a few special notes or the special considerations for the iRECIST. And for the last 1 minute, we'll be briefly talk about what we are as a Merit and what we do. Starting off with the iRECIST background and the purpose. The background is basically iRECIST came out as a modification of the RECIST 1.1, which is the most commonly used response criteria for solid tumor, but the iRECIST is a slight modification to accommodate the immunotherapy drug that causes the pseudoprogression of the tumors. But still the fundamentals and the purpose of the iRECIST is the same. It provides the objective methodology to assess the tumor responses to the medication or the investigational drug that is being used for clinical trials. And indication again would be the same as a RECIST 1.1. It is a solid tumor, but treated with the immunotherapeutic agents. And speaking of the pseudoprogression that is commonly seen for any medications that uses the mechanism of the PD-1 or the CTLA-4 inhibitions, pseudoprogression is defined as an increased size of the lesions or any visualization of the new lesions that could indicate the progression of the disease, but also after that followed by a response. So which means the pseudoprogression, denoting that it was not like a true progression, but it eventually resolves all the symptoms and the lesions. It ranges approximately 6% and does not go over 10% from the most of the studies in the subjects treated with the immunotherapy. But the iRECIST wants to make sure that it is the response criteria accounts for any pseudoprogression that may lead to a response at the end of the day, just to make sure that treatment is not prematurely stopped for those people who may have a chance to have a response after pseudoprogression. For the iRECIST, most of the standards remains the same as the RECIST 1.1 with the slight modifications. And these are the list of the standards or the definitions that still remains the same. So that would be measurability definitions, and also the target, non-target lesion requirements and the definition. How we handle the nodal lesion still stays the same with the measuring the short axis. And also method of the measurement, UP unidimensional, that stays the same. Also, the sum of diameter is one of the quantifications that we still use as the same way. Also, the CR, PR, SD definition stays the same. Confirmation of the complete response and partial response also stays the same. And then lastly, the definition of the progressive disease, which is used as an IUPD in iRECIST, that still stays the same. But there are a few new concepts or added algorithms or rules that's added to iRECIST on top of RECIST 1.1 is how we handle the new lesions. So the new lesions now is categorized into two different categories of the new lesion target and also the new Lesion-Non Target. And then the time point response after the initial PD is handled slightly different. So there is a concept of the progressive disease as an unconfirmed progressive disease and a confirmed progressive disease. And also additionally, collection of reason why the IUPD or the unconfirmed progressive disease have never been confirmed. Those type of reasons are also collected through the ECRF. And these lists of the reasons can be predefined per charter or the protocol. So getting into a little more details in iRECIST, measurability of lesions still stays same as the RECIST 1.1, but I will be briefly going over and touch base some of the overlap details of the iRECIST and RECIST 1.1 here. So the definition of the measurable lesion still stays the same. We would use the unidimensional measurement of the long axis of the non-nodal or the short axis of the nodal lesions. And the longest diameter of the lesion has to be at least 10 millimeters by a CT or MR scan with the 5 millimeters or less slice thickness. And also the nodal lesions has to be at least 50 millimeters in short axis by CT. So these measurability requirements still stays the same. And also the definition of the non-measurable lesion is also the same. Any other lesions that do not meeting the requirements for the measurability, which is all small lesions, or any lymph node that short axis runs between 10 to 15 millimeters. Any truly non-measurable lesions, which are listed down here, such as ascites or pleural effusions, those type of lesions are considered as a truly non-measurable just due to their presentation type or the morphological feature. These are also considered as a non-measurable lesions. And also the method or the recommended modality of the capturing the lesion would still stay the same. CT or MR with the proper consistent imaging parameters is recommended. And other modalities are still will stay ancillary and may not drive any type of responses in both RECIST and 1.1, which would include the clinical exams such as a caliper or the chest X-ray, ultrasound, endoscopy. These type of things are not recommended to be driving the objective response for both RECIST 1.1 and iRECIST. And for lesion category, now instead of 3 categories, now we will have a 4th, which is the target lesions. We use a quantitative assessment, non-target lesions, qualitative assessment. These two will stay the same as RECIST 1.1 in iRECIST, but now new lesions is now divided into the 2 delineation, which is a new lesion target, which we'll be using a quantitative assessment within these lesion group, and then a new Lesion-Non Target, which is a qualitative assessment. So there will be 4 different categories of the lesions that we would categorize all the tumor burden of the patient. So firstly, target lesions, which also stays the same as a RECIST 1.1 in the iRECIST. We still have we will still follow the same requirements or the guidance of the maximum of 5 lesions per subject, which has to be measurable lesions, which should mean longest diameter of 10 millimeters or greater of the non-nodal, and short axis 15 millimeter or greater in the nodal. No more than two lesions per organ still stays the same. And then selection criteria for target lesions are also the same, which should be based upon a size and a reproducibility. And those target lesions will be representative of the organs or the subject or the disease type at the baseline. Assessment of the target lesions are also the same. Baseline, we will measure lesion measurement in millimeters. Also the lesion state as a qualitative assessment of the being present. Anatomical locations, laterality, directionality, and the sum of diameters will be calculated upon a baseline visit. Upon follow-up visits, each of the lesions identified in baseline will have another measurement of each lesions, and also the sum of diameters will be calculated for deriving the response of the target lesions. Also, the tumor state will be recorded for the qualitative assessment. And in any cases, lesions are getting too small, we can record the too small to measure, just like how the RECIST 1.1 has been done. For the non-target lesions, any measurable lesions that did not get included in the target lesions at the baseline will fall under all non-target lesions. And then any of the truly non-measurable lesions. We would still follow the same rules for RECIST 1.1. The size measurement is not required and it's being an optional. So for the non-target lesion assessment for iRECIST, it would be still the same as a RECIST 1.1. At the baseline, we'll be recording a location of the lesions and a lesion state and the size as an option. And upon a follow-up, we will record how the tumor has progressed or responded, basically absent, present, or progression. Same for the nodal, whether it still stays the pathologic or non-pathologic, or pathologic but progression. And also the not evaluable could be an option to record in just in case scans has a bad quality or missing visits. Getting into the new lesion target, which is a slightly different requirements compared to the RECIST 1.1. Now, any new lesion that appears upon a follow-up visits will be assessing what new lesions will fall under the target lesions. And here are some requirements for the new lesion target, which looks very similar requirements as a target lesion upon a baseline. So new lesion target can be only up to 5 new lesions that are measurable, and it should be no more than 2 per organ. And the longest diameter of the non-nodal short axis is the lymph node lesions. So that's very similar to what the target lesion at the baseline behaves. Also, the size requirements still stays the same of the longest diameter of 10 millimeters or greater. If it's a nodal lesions, 15 millimeters or greater on their short axis. And also the new lesion target will be recording a sum of diameters just for the new lesion target, which is separate from the sum of diameter of the target lesions. So each new lesion targets will have a measurement, either measurement or the too small to measure or not available as we assess further time points after the new lesion has appeared. And also for the new lesion non-target, this would also follow the very similar requirement as a non-target at the baseline. So anytime the new lesion has appeared upon follow-up, and if there were any lesions that were not included as a new lesion target group, will be falling under this new lesion non-target. This will follow the qualitative assessment of the absent, present, increase, and not evaluable. And these can have a further delineations we will be discussing later in this discussion. So in a summary of the lesion category, The baseline tumor burden is first identified as more of the measurable or non-measurable lesions, and now they can be grouped into the target and the non-target lesions. So that will be a patient's baseline tumor burden. These lesions within a target and a non-target group will be still being followed upon any follow-up time points. And then upon a follow-up to time points, we'll be assessing if there's any new lesions that will fall under the new lesion target, new lesion non-target. So there'll be a 4 group of the lesions that will be being followed at the follow-up visits. For the iRECIST response criteria, definition of the CR/PR/SD and also the NN, the non-CR, non-PD for the non-target response will stay same as the RECIST 1.1. But just the nomenclature has slightly changed it over here, which in RECIST 1.1 they would denote these abbreviation as a CR/PR/SD and NN. For iRECIST, we just add letter I's in front of each of the abbreviations just to denote the letter I for iRECIST or the immunotherapy. And for the iRECIST response criteria, the main difference of how we assess the time point response changes when the patient first initially shows the signs of progression in the CT or MR in the radiologic assessment. And here in the diagram, upon a progression is seen at the visit, if RECIST 1.1, it would simply be assigned as a PD. But initially, whenever the progression is seen at the visit, when we read for the iRECIST, we first assign the IUPD, which is the unconfirmed progressive disease. Unconfirmed progressive disease, or IUPD, may occur multiple times, such as an example would be an ICR, and the next time point patient can have an IUPD, and then have another IUPD, or something like IUPD, and then patient resolve back to IPR, and then reoccur with the IPD again. So unconfirmed progressive disease may occur several times. And the definition of the IUPD is still same as the RECIST 1.1 PD, which should be a target lesion. Sum of diameter increases 20% or more from the nadir or the baseline, and also the at least 5 millimeter of the absolute increase together, or the non-target lesions that reader assesses that this is an unequivocal progression, or any new lesion appearing. These 3 conditions may occur concurrently, or only one lesion group, which would cause a PD. In iRECIST, it will be an IUPD. And then for ICPD, which also is a new concept compared to the RECIST 1.1, is a confirmed progressive disease. A ICPD, it only comes right after the IUPD has been assigned to the subject, and usually the confirmation period is about 4 to 6 weeks from initial IUPD. There is a specific requirements or the conditions that will trigger the ICPD for whoever the subject had an IUPD in an immediate prior time point. So basically, from the initial progression, we assign an IUPD, and depending on how patients respond in the next time point, if the conditions for the ICPD has been met, then at the next time point, we assign an ICPD saying the progression of prior time point has been confirmed, and that would be denoting a true response. If the lesion category assessment has shown not meeting that ICPD, then patient can take a different path of their time point response in the next time point, which patient may have an options to remain in IUPD, or if there is any responses or resolution of the lesions that could lead to the ICR, IPR, or IST, which is getting out of the unconfirmed progressed disease, which is a scenario for accommodating for the possible pseudoprogression, which should be this path we'll be talking about pretty soon. Firstly, I want to talk about the case where the ICPD can be assigned. So there will be two cases or two conditions that ICPD will be assigned to a subject immediately after the IUPD has been assigned. So the first scenario is if the original cause of the IUPD, which is one of the lesion or the one or more of the lesion category that caused the IUPD in one time point, worsens in a subsequent time point. And here's the definition of what the worsening of that, further worsening of that lesion category that will cause the ICPD. So, and also these can happen in a single lesion category or the multiple lesion category at the same time. So we discussed that the target lesion can cause an IUPD for the RECIST 1-1PD definitions. If that has caused, and right after that time point, if the target lesion further worsens in this way, which should be a sum of diameter increasing 5 millimeters or more, that will cause the ICPD at that time point, being an target lesion being the original cause of the IUPD, and then further worsening of the 5 millimeter or more in SOE. For the non-target lesions, if the non-target lesion has caused the IUPD in original time point, which is an unequivocal progression, at the next time point, same non-target lesion group, if there were any increase that's seen as a quality assessment by the reader, that is a conditions to trigger the ICPD in the next time point. For the new lesion target or new lesion non-target, the appearance will initially give the IUPD on one time point. Immediate after this time point, if the new lesion target sum of diameters increases 5 millimeters or more, that is a conditions to give an IUPD, ICPD due to the new lesion target, or if there were any increase qualitatively assessed by the reader, or if there's any further new additional lesions on top of the new lesion that has been appeared in the original time points are seen, those will be conditions for a ICPD. So that's the first scenario where ICPD can be assigned. The second cause or the second conditions or scenario where ICPD can be given is that if the lesion category that did not originally cause the IUPD in a prior time point, which is the one immediate prior time point, now meets the requirement or the RECIST 1-1PD on the current time point. So the examples would be, for instance, on a time point 1, if the target lesion was the only lesion category that has caused the IUPD, and the non-target or the new lesions did not cause any issues on a time point 1 for the IUPD. On a time point 2, target lesions may stay the same, but if the non-target lesion now meets the RECIST 1-1PD requirements, which is an unequivocal progression, or there were no new lesions in the time point 1, now we have a new lesion appearing that will lead to the ICPD. So basically, at the time point 1, target lesion was the cause of the ICPD, but not the non-target or the new lesion. Time point 2, now problems are arising in the non-target or the new lesions with the condition of the RECIST 1-1PD requirements. That will give the ICPD upon the time point 2. Another lesion would be upon a time point 1, new lesion was the only lesion category that has caused the IUPD. So basically, time point 1, we found a new lesions that will give the IUPD. Target and non-target did not cause any issues for the progression on a time point 1. Now, upon a time point 2, target lesion sum of diameter increases 20% or more from the baseline or nadir with minimum 5 millimeter increase and or non-target lesions unequivocal progression. So basically, in this case, the non-target target lesions did not cause an issue on a time point 1, now meeting a RECIST 1.1 PD requirements, which has to be 20% increase, not just simply only the 5 millimeter, which would be first cases. So that would be a second scenario or the second conditions where ICPD can be given to the subject. And then after we discussed the two cases where the progression can be confirmed and giving an ICPD as a time point response for the subject, we have a different path down here. Patient may still stay in the IUPD or may get out of the IUPD status. So if the requirement for the ICPD did not occur, following options can happen for the patient, which is patient still staying in IUPD, so just assigning another IUPD. This would be a more like patients being stable upon initial progression. Basically, if the ICPD requirements did not occur in any lesion criteria, so the ICPD were not given. And if the target lesion is still in a PD range, basically the target lesion did not resolve at all, so still in an IUPD range, then the patient will stay in the IUPD. And then further follow-up time points will have to be assessed whether patient will take a different path out of the IUPD, or they may still be in an IUPD. For IUPD, goes to the ICR, is when initially IUPD has been seen in a time point, and followed by all the disappearance of the lesions in all lesion categories, then the ICR conditions can be met and patient can get out of the IUPD and then be in a response status. Also, there is cases where patient might resolve up to the partial response or the stable disease area, which the target lesion is below the PD threshold, so basically in the range of the partial response or the stable disease range from the IUPD. And also the non-target lesions and new lesions are improving or stable. This would also mean that it's not causing any ICPD, that next time point would not be ICPD. So here are some examples. On a time point 1, the subject has a baseline sum of diameter of 100 millimeters. New non-target lesions are present, and the new lesions, there's no new lesions on the baseline. Visit 1, we have a stable SOD, or the sum of diameter, stable non-target lesions. So we give INN. This will lead into the stable disease in the time point response. At one visit 2, target lesion has increased more than 20% in their sum of diameters, and the non-target still stays the same. No new lesions has been seen. So because of the target lesion, we will assign IUPD as a time point response. And the next time point, we have a case where the only a 1 millimeter increase has occurred with no much change in a new lesion or non-target lesion. So basically, this is less than a 5 millimeter. So this is not conditions to cause an ICPD. So it will stay in an IUPD. Upon visit 4, the target lesion still stays the same, but the non-target lesion now meets the unequivocal increase, which is the RECIST 1.1 PD definitions. Now this would be giving an ICPD because of the non-target lesions, and this would be the second scenario where subjects are given with an ICPD that we earlier discussed. There are another examples here, very similar to the earlier ones, with the new lesion causing issues in this subjects. So same scenario, baseline status is the same. On a visit 1, we have a stable disease on our target lesions with no much change on the rest of the lesion categories, which would be ISD. Now on a visit 2, target lesion sum of diameter still is in a stable disease range, but now we have a new lesion target has been found with the sum of diameter being 10 millimeters. So that would be an IUPD because of the new lesions. For next time point, visit 3 and 4, still the target lesions are within the stable disease range, but the new lesions are also no much change, only 2 millimeter increase across the 3 and 4 visits, which is not a 5 millimeter increase in a new lesion target sum of diameter that cannot cause the ICPD in any of the visit 3 or 4. But now we're seeing that additional new lesion has been found that were not seen in any of the prior 2, 3, 4 visits. So this is an ICPD that is triggered by the new lesion category due to the additional new lesions. And then also for oncology trial imaging endpoints, RECIST or iRECIST or any response criteria utilizing the imaging response criteria has a very similar way of handling the imaging endpoints, which the two main group is the tumor response rate endpoint, often referred to as an objective response rate. This would be a proportion of the patients meeting complete response or the partial response out of the all subjects being included in the trial. Also, the time to event endpoint can be utilized for a progression date or the PD date. And usually the progressive-free survival is the one of the endpoint often used with the RECIST or the iRECIST. And this is defined as a time from the randomization until objective tumor progression, which is the PD by the RECIST or iRECIST, or the death, whichever occurs first. So that would be a two very common endpoint being used for the RECIST 1.1 and then iRECIST. And then for the iRECIST, there are some non-radiologic data that can be considered to be collected. Recommended information to be collected per iRECIST is that if there were any reasons that patients were in the IUPD throughout their visits and never has been confirmed, there could be a protocol-defined list of the why IUPD has not been confirmed to the ICPD. And also there are additionally clinical status the patient can contribute to the progression as well, which is the any subjective or qualitative clinical status assessment by the individual provider treating each subject. And those assessments can be related to their performance status or any tumor-related symptoms. As of the ECRF, in the iRECIST should be collected as a two category data, such as a clinically stable and unstable. For some special consideration for iRECIST, we have a data progression that may look slightly different from the RECIST 1.1. Apologize here. So the progression date will be backdated to the progression. The original progression date where IUPD has occurred that has led to the ICPD. So such as an example here is subject starting with an stable disease with the IUPD, getting out of the IUPD. So the subject now has a stable disease. Now they went back to the IUPD, and finally that IUPD has been confirmed as a confirmed progressive disease. In such cases, the date of the progression will be backdated to the time point for where unconfirmed progression was the initial progression that was seen. Eventually that has been confirmed. So time point 4 PD date will be used for the progression analysis. And also some other cases where is responses after IUPD due to the new lesions. So because of the pseudoprogression and accommodation of the pseudoprogression, there are still presented new lesions still resulting in a response in a time point overall response. So in that cases, new lesion is stable and their size is not causing a ICPD. So in such cases, subject may get out of the IUPD and still in a IPR or ISD, with the new lesion being stable. In such cases, these are the conditions that patient may go back to IUPD due to the new lesions, which should be cases where patient have a time point response of the IPR or ISD with the stable new lesions and a subsequent time point if there were additional new lesions found at that time point, or now the new lesion target following a RECIST 1.1 criteria in their sum of diameters, which is their 20% increase, or the new lesion non-target meeting a RECIST 1.1 criteria of the PD, which should be a unequivocal increase, or overall 73% of the increase denoted by RECIST 1.1. These cases can cause the IUPD from stable new lesion with the response in that time point. Also, for another considerations in a non-target lesion assessment is that there could be a little bit more of the delineation, more than just simply a absent, present, and progression. The iRECIST recommends that 2 additional type of the quality assessment can be recorded in an ECRF, which should be a no change from the baseline, increase from the baseline or the nadir, and then also the additional one is a no change from the last assessment or the further increase from the last assessment, so that these 2 type of the qualitative assessment can be utilized whenever new non-target lesion has caused the IUPD, and whether these are actually non-target is actually whether triggering the ICPD or not. These 2 options of the qualitative assessment in the ECRF can actually help making more clear documentation of whether non-target lesion is really causing the IUPD in such cases, and also the unequivocal increase anytime these type of the progression is seen in non-target. So examples would be, for instance, at the baseline, we're starting off with the 100 millimeter of the sum of diameters in target lesions. New non-target lesions are present and no new lesions. Time point 1, we have a slight increase in sum of diameter, which should be a stable disease, and a no change in non-target lesion. We're using a no change, which is on INN, and then none for the new lesions. So that adds up to the stable disease in the time point response. Now for the target lesions still being stable, but not causing a PD, but now we use one of the documentation way of the unequivocal increase with the the new lesions. So that's an IUPD because of the non-target lesions, so that the TP response adds up to be an IUPD. And upon time point 3, there's no much change in the target lesion again. Also, we're using this way of the documentation that no change from the last assessment. That denotes that from the unequivocal progression of the non-target lesions, there was no further increase. So this denotes that this is not a case for the ICPD, but the non-target lesion is being stable post the IUPD. So that adds up to still patient remaining in the IUPD status for that time point 3. Different cases would be increase from the last assessment, which should be a different route in time point 3 for different subject, where now the unequivocal non-target lesion shows a further increase from the last assessment. So this denotes that from the unequivocal progression, there was a slight more increase. And this INCLA will be noting that it's causing an ICPD because of the non-target lesions. Also, another thing to consider is that iRECIST is still not validated response criteria. So the RECIST 1.1 is still being a standard and then well-accepted criteria for the solid tumor. And then iRECIST often becomes the exploratory endpoint still. So this is the recommendation from the iRECIST that they recommend that iRECIST uses as an exploratory endpoint. And the data collection of the iRECIST still includes all the data required for the RECIST 1.1. So whenever the iRECIST is read, you can extract the analysis data that's needed for the RECIST 1.1. So it is preferred that using a single workflow for iRECIST and then also remap the final data to the RECIST 1.1 so that the one read can eventually present the two different response criteria endpoints, and both of them are recommended just per iRECIST publication, just to help how the RECIST 1.1 and iRECIST will compare in the same study, and also provide a further validation of the iRECIST usage. Also, there are a few common mistakes in iRECIST, which is sometimes the nadir gets a little confused occasionally, and that is because of the concept where patient can get into the IUPD and then be out of the IUPD into the response status, which would be such as an ICR, IPR, or ISD. Even though patient gets out of the IUPD, that does not really mean that the subject is rebaselined after they got out of the IUPD. So that means always the baseline is the initial tumor burden of the baseline visit, not any new values that's given after the IUPD is or the patient got out of the IUPD. So that also means that the nadir is always the nadir, the smallest sum of diameter from entire visit that is available. So it will be a smallest sum of the diameter visit in a global time points. And then also another thing is that ICP from the IUPD by the non-target lesions. And in this case, if the non-target lesions or the new lesion non-target has caused the IUPD, and when a ICPD is assigned a next time point, it'll be any further increase, and it has no need to be an unequivocal increase. So any slight increase can cause ICPD from IUPD in case of the non-target lesions or the new lesion non-target groups. Also, the one other thing to remember is that target lesion response still weighs more. So basically, such cases we discussed earlier, after IUPD, if the target lesion becomes ICR, IPR, or ISD, and if the rest of the lesion categories are still in the same status or no much change, the target lesion outcome can derive the overall time point response, which should be, we have a short example here. We have 100 millimeter SOD in the target lesions upon baseline, stable new non-target lesions at baseline with no new lesions. And if the time point 1 target lesion sum of diameter becomes 110 millimeters with no change in non-target, no new lesions, that will be unstable disease in a time point response. And next time point, non-target lesion has shown unequivocal progression that caused the IUPD. And as far as the next time point, if the non-target lesion has not really caused the ICPD, if they are stable or if they're improved, and if the target lesion has tremendously improved, that they are down into the partial response range, in such cases, if none of the other two lesion criteria has caused any worsening or the ICPD requirements, the target lesion response can drive the overall time point response. In such cases, this would be a IPR in the TP response of the time point 3 due to the target lesion being improved with these other two lesions not worsening in other way, meaning the ICPD has not caused by any of the other two lesion categories. And I appreciate for all the attentions for our discussion today, and I will hand my presentation back to Amanda.
Thank you, Jean. Great timing, right at 1:45. We have reached the Q&A portion of the presentation. If you have any questions, please submit them now via the Q&A functionality. And prior to addressing those questions, over the next 2 slides, I would like to briefly highlight Merit's associated capabilities. Merit is an innovative global clinical trial endpoint services provider focused on a variety of therapeutic areas while specializing in ophthalmology, respiratory, and oncology. We have served biotech and pharmaceutical companies in their clinical endpoint analysis for over 10 years with a team of medical professionals as well as seasoned operational support staff. Positioned as your clinical endpoint expert, Merit provides comprehensive endpoint assessments from acquisition directly from the sites, storage, quality reviews, and expert analysis of that data to be then exported and returned to the sponsor, all within a collaborative, partnership-driven environment with customized solutions and personalized attention. Our proprietary software platform, Excelsior, delivers automation in a single system for all image and workflow management, resulting in clean, complete, and accurate data that will meet your clinical trial needs. Key imaging software features include the interactive and semi-automated response criteria reading forms, which provide cost-efficient reader workflow, as well as minimize risk of bias assessment with independent blinded review between readers and adjudicators. We will now open the discussion for any questions that you may have. All right, we have our first question here, Dr. Kim. We are transitioning from preclinical to clinical studies. How do we get started with imaging endpoints?
Thank you very much for the question. So from the preclinical to the clinical, first 2 things to consider is that what are the target disease states, whether it's a solid tumor or whether it is a liquid tumor such as lymphoma or the other indications. Those will actually firstly derive what response criteria to be used in a phase 1 study such as RECIST 1.1 or renal or Lugano for other indications such as a glioma or lymphoma. Also, we need to consider whether the mechanism of the investigational drug includes any of the potential pseudoprogressions, such as utilizing PD-1 inhibitions or immunotherapy class is being used for phase 1. Then we would also need to consider the pseudoprogression cases. It's in such cases, as we discussed, the iRECIST can be utilized as an exploratory for solid tumor, but also there are the immunotherapy versions for different tumor types. Glioma would have an iRANO, and also a lymphoma would have a lyrics as on their version for immunotherapy.
Okay, great. Thank you for that additional insight. We have another question for you, Dr. Kim. Do sponsors ever want to change the iRECIST criteria?
I could see there could be a request and consideration for the iRECIST modification as well. Just like a RECIST 1.1, we do occasionally make some modifications on target lesion requirements, such as a maximum of 5 target lesions, no more than 2 per organ per subject. Those type of the modifications still can apply for the iRECIST per sponsor's request. Also, we can have a slightly different requirements of the anatomical locations allow for the target lesions versus non-target lesions. Such can be used in the RECIST, also can be reapplied to the iRECIST as well. So those modifications, if they have a good rationale that is brought from the preclinical studies and other evidences, that could be totally a possibility for making a modification to the response criteria.
Okay, thank you. We have another submitted question. In determining IUPD 100 millimeters to IUPD 102 millimeters to 105 millimeters for target lesions, would this third time point by ICPD from the 5 millimeter increase from the original IUPD?
Yep. So the 5 millimeter increase would be a either can be a one instance of the 5 millimeter or more increase, or it can be increment of the total 5 millimeters across multiple time points.
Okay, great. And I think in respect for everyone's time, we will address one last question, Dr. Kim. What key elements are collected in the read ECRF form during an iRECIST read?
Okay, thank you for the question. For the iRECIST, the key elements for the ECRF would be firstly, lesion ID for every lesion that is being identified in baseline, basically target lesion and non-target lesions. They should all have the lesion IDs and associated annotation on the scan. And then also the measurement in millimeters for each target lesions, and then also the tumor state, a qualitative assessment record for the target and non-target lesions, sum of diameters, and a response for each time point is also required to be annotated or they recorded in an ECRF. And then also for the new lesions at any follow-up time point, they should also make sure that each new lesions will have the new lesion ID and their associated measurement values and qualitative assessment. And then also it is important to make sure that assessment date or the new lesion identification dates are also recorded for multiple new lesions being occurring across the different time points. And that should follow the actual procedure or the modality date of the CT or MR scan that's being submitted for that visit.
Okay, great. I think we've covered a lot of informative information. So thank you, Dr. Kim. We have now reached the end of the Q&A session. Thank you once again to all of our attendees for your time today. You can stay up to date on additional primary endpoint related content by following our company page on LinkedIn. And please stay tuned for more information regarding Merit's upcoming webinars and have a great day.
Thank you.